Chloroplast DNA. Like the mitochondria have their own DNA, chloroplast as organelles also have their own DNA situated within the stroma. Now, interestingly, what has been observed is that chloroplast DNA seems to have a lot of similarity with the genome of the cyanobacteria. And therefore, there is a suggested hypothesis again that this chloroplast is an endosymbiont which has come from a cyanobacteria. Therefore, for a cell which holds chloroplast within it, so for example, plant cells, certain fungi, or algae, these all will have, apart from the nuclear DNA, it will have chloroplast DNA as an extra chromosomal DNA, and most of these cells will also have mitochondria. So therefore, there seems to be a network that is existing between the nuclear DNA, the chloroplast DNA, and mitochondrial DNA, and together they are taking care of several functionalities of these cells. So let us look at the outcomes of this session. The chloroplast DNA is a closed circular covalent DNA. Mostly. Now there are a lot of evidences suggesting that it need not be a closed circular covalent DNA. It contains genes that helps in the process of photosynthesis. The chloroplast genome consists of multiple copies with genes coding for essential proteins, rRNA and tRNA. There is transfer of chloroplast DNA to the nucleus and vice versa. So what has been observed is a very interesting phenomenon of genes from the chloroplast DNA moving to the nucleus, while genes from the nuclear DNA moving to the chloroplast. And one can also observe that happening with the mitochondria. Although what has been observed over a period of time is that there is more of movement of genes from the chloroplast to the nucleus, and therefore this makes chloroplast DNA genomics an interesting area of study. So basically what has been understood is that the chloroplast DNA is still evolving, the chloroplast genome is still evolving. Studies have shown that chloroplast DNA are engineered for biotechnology applications. Let us look at and understand what is present in the chloroplast DNA. The chloroplast DNA is much larger than the mitochondrial DNA. And what has been observed is, if you consider that the chloroplast DNA is a closed circular structure, closed circular covalent structure, then it is observed that it can range from anywhere between 120 kb to 247 kb, and there are other variants too. So the size can vary from species to species. And interestingly, what has been observed is that the number of chloroplast DNA within chloroplast can also vary from 22 to 900 copies. So, for example, in response to light, it may be possible that the number of copies of the chloroplast increase under certain conditions. So, this variability in the number of chloroplast DNA within a chloroplast also is dependent on the chloroplast function itself, whether it is carrying out more of photosynthesis or whether it is responsible or carrying out nitrate or sulfate a homeostasis, or if it is basically involved in counteracting certain stress. So depending on what the plant or the algae is undergoing, you would have the number of copies of the chloroplast DNA altering. So therefore, it is understood to be a need-based replication, and chloroplast replisome machinery has been observed to replicate, including with proofreading. And therefore, it has been observed that there are variations, and if there are variations, then it is going to basically affect the phenotype of the plant or the algae. So the genotypic change in the chloroplast has been observed to have a phenotypical effect. The chloroplast DNA contains somewhere around 120 genes, and a few chloroplast genes have introns. Now, this is an extremely important find because generally one looks at the fact that, if you consider that chloroplast DNA has evolved from cyanobacteria, then cyanobacteria as a prokaryote would not have genes containing introns. Like we know that mitochondrial DNA, the genes do not have introns. And thereby, this connect of it having evolved from prokaryotes and prokaryotes not having genes with introns, that lies true over here. So, here where you find chloroplast and you find that genes have introns, this spikes the interest in the fact is how does the chloroplast DNA have genes with introns if it has been evolved from, or if it has come from a cyanobacteria. So here is where the interesting study of exchange or transfer of genes from nucleus to mitochondria, mitochondria to chloroplast, chloroplast to nucleus is is understood to be happening, and therefore there is a possibility that few genes have got transported from the DNA of from the DNA in the nucleus to the chloroplast per se. But these things are being studied, and over a period of time there will be more clarity on this phenomenon. 4 genes are for ribosomal RNA, 20 genes are for ribosomal proteins. So we all know that chloroplasts also have their own ribosomes, and genes that have been transcribed from the chloroplast DNA are translated within the chloroplast itself. And therefore, the ribosomes that are formed, many of the proteins that assemble into ribosomes along with the ribosomal RNA are formed from the genes present in the chloroplast DNA itself. But please mind it, not all, because ribosomal proteins are 52, more than 50 proteins form the ribosome. So the rest of the proteins come from genes in the nuclear DNA. So that is definitely the case. So it is something that has been observed in mitochondria also. In mitochondria, not all functions take place just because of the genes present in the mitochondrial DNA. Similarly, in chloroplast, again, you do not have all genes present in the chloroplast DNA. Many proteins that function in the chloroplast have come from the nucleus per se. There are 30 genes for the transfer RNA because we know that for the synthesis of proteins within the chloroplast in the ribosomes that are formed, tRNA are essential, and 30 genes for tRNA have been observed in the chloroplast DNA. There are obviously genes that code for proteins involved in photosynthesis, not all of them, but many of them. There are 6 genes out of the 8 ATPases that are present in the chloroplast. 6 ATPases are coded by the chloroplast DNA itself. There are genes for RNA polymerase. These RNA polymerase specifically transcribe the genes that are there in the chloroplast DNA. And it is interesting to note that the large subunit of Rubisco is encoded by the chloroplast DNA, while the small subunit of the Rubisco is coded by the nuclear DNA. So, for the functioning of the Calvin's cycle itself, for carbon dioxide fixation itself, you can see what the contribution of chloroplast DNA is, because Rubisco, that is ribulose bisphosphate carboxylase oxygenase, will not be formed if you do not have this gene in the chloroplast DNA. So, therefore, it plays an essential role in photosynthesis per se. Moving forward, there is an ongoing process of transfer of genes from chloroplast DNA to nuclear DNA and also mitochondrial DNA, an aspect we just understood or we just contemplated when we saw that there are several genes in the chloroplast DNA that have introns. It is also, therefore, a contention at this point of time that chloroplast DNA has not completely evolved. It is still in the process of getting evolved. there are more genes from the chloroplast DNA getting transferred to the nucleus. Now, the chloroplast DNA also has been observed to have several repeating DNA sequences. So, when we talk about repeating DNA sequence, it is a sequence which you will find more than once in the DNA. So, there are actually about 6 families of repeating DNAs that are found in several chloroplast DNA. There are 2 inverted repeats. This is something that has been found in a lot of chloroplast across species. Uh, so, for example, what is observed is that you have an inverted repeat A and an inverted repeat B. When you say inverted repeat, what does it mean? This sequence is this side and this sequence is this side. So, it's not, the sequence is not the same side, the sequence is inverted. And interestingly, the positioning of the inverted repeats are such that you have a long single copy and you have a short single copy. So the chloroplast DNA itself is divided into, 2 major parts, with this being one and this being the other. So, but again, please do note that it is not necessary that you always have the inverted repeats positioned likewise. But majorly, it has been found to be positioned likewise. There are also 2 tandem repeats. So, tandem repeats is, if you have a repeat sequence here, you have a second repeat sequence here, and the third and the fourth. So, it keeps repeating almost immediately. Then you have 2 dispersed repeats, that is, they are present anywhere. Now, these 2 inverted repeats have found to be studied a lot, and it seems to be anywhere between 10 to 80 base pairs and more also sometimes. They have been observed to be conserved sequences within species. All and Many of these sequences, that is, the inverted repeats, contain the rRNA genes. So sequences may vary, the conserved, that is these inverted repeats, the sequence may vary from algae to plants. So between species, they have been found to be different. Not just different in terms of sequence, but also in terms of positioning. So it's not necessary that they are positioned in the same way. So those things are done. Now, another aspect over here that becomes important is because these conserved sequence contain the rRNA genes, therefore rRNA of even the chloroplast can be used for identification of certain plants or for identification of certain algae, etc. Moving forward with chloroplast genomics, there are now when we use the word genomics, it is basically studying the genes present in the DNA. There are evidences of the chloroplast DNA also existing as linear DNA. Now, this has been something found to be very interesting. Why? Because it has been observed as those chloroplasts that have only a single inverted repeat somehow have been found to exist as linear DNA and also have been found to be broken into 2 parts. So, one conclusion that has been made by several studies, but again, it is not a final conclusion, that it may so happen is that if you remove the inverted repeats from the chloroplast DNA, there can be more breakage of the chloroplast DNA, or there can be more rearrangements of genes within the chloroplast DNA. Studies, as mentioned here, they in absence of the inverted repeats, they can undergo more breakage and rearrangements. NCBI has over 800 chloroplast DNA sequences present as data. That means that more than 800 chloroplast DNA from different species have been completely sequenced, and the annotation of those sequences are present as databases. So, therefore, the genomics becomes, therefore it is called as genomics because you have all the data for different genes present in different chloroplast available as a database, which can be used for analyzing and studying lots of aspects related to chloroplasts. Now, due to the sequencing, okay, and using the new gen sequencing, it has been found that chloroplast DNA does show variations not just within species as well as between species. And These variations seem to help the plants adapt to various climatic changes. These variations also give an insight into transfer of genes to nucleus and the mitochondria as explained earlier. And this also kind of basically refers to the fact that the variations can enable possible tolerance to certain stress conditions or adaptations to certain stress conditions, and these adaptations happen because of changes in the chloroplast genome. Therefore, having understood these variations, one could one could actually use these known variations as means of improving crops. So one can actually bring in those changes in the chloroplast genome to get better crop. So that is something that has been taken up by many scientists across the world to use chloroplast DNA as a means to improve crops. DNA barcodes enable identifying specific varieties and then can be conserved for breeding. What do we mean by DNA barcodes? Well, suppose you have bamboo and there are several varieties of bamboos. Now, each variety of bamboos within its chloroplast DNA may have a sequence very unique to only that variety. So therefore, that becomes a barcode. That means that that's an identity for a particular variety. And so, if one sequences, if one observes a certain bamboo plant and one wants to find out what is the variety, and one isolates the chloroplast DNA, sequences it, and if one finds a sequence which is very similar to a barcode of a certain variety, one can actually identify that variety. Okay, so that is one thing that has definitely evolved over a period of time. Desirable traits from unrelated species can now be readily introduced into commercial cultivars. So you can actually carry out transgenesis, and with transgenesis, therefore, several transplastomic plants have been created. This word is used more only and only if the chloroplast genome has been either altered or a gene of interest has been inserted into the chloroplast DNA. So in fact, over here you may look at the fact that having known the sequence of chloroplast DNA, one is using the chloroplast DNA as a vehicle to transfer a gene of interest into a certain plant type. Say, for example, we are taking only one example, but you can find many examples. So, for example, production of a subunit vaccine candidate against porcine post-weaning diarrhea in high biomass transplastomic tobacco. That means a tobacco has been created, okay, by inserting a gene in the chloroplast DNA. So, this you can see is a plasmid that has been constructed such a way that it can integrate into the chloroplast DNA. And this construct has a gene that is going to give rise to a protein that can act as a vaccine for the pig. Against what? Against diarrhea. Okay, so therefore you can see how once the construct has been applied and you have the necessary genes present within that gives rise to the vaccine, okay, that gives rise to the recombinant product. And you can see over here, if you have a wild type plastome without the gene of interest present, then this is only about 1054 base pair. But if you have introduced this plasmid chloroplast into gene construct into a chloroplast genome, then it would be anywhere between it would be about 3000 base pairs. So with the gene of interest, it is 3000 base pair. Without the gene of interest, it is only 1054 base pair. So once this has been introduced into the chloroplast, and if it gets integrated within the chloroplast DNA, then as one observes, this is plant 1, plant 2 and plant 3. And you can see over here how plant 1 has the vaccine, plant 2 has the vaccine, But plan 3, which has not been introduced with the transgene, is about 1054 base pair only, and therefore this is not producing the vaccine, but these two are producing the vaccine. So these plants that have been created are transplastomic tobacco. So let us make the conclusions. Chloroplast DNA comprises about 123 genes coding for proteins related to photosynthesis and transcription, tRNAs, and cpDNA has been observed to have inverted repeats whose position and sequences show variations within species and between species. Sequencing of the chloroplast DNA of various species has created a database that enables chloroplast genomics research, especially for crop improvement, evolutionary studies, and transfer of genes from chloroplast to nucleus and mitochondria. Using chloroplast genome constructs, several transplastomic plants have been created for various applications such as production of recombinant products, biofuels, etc. Chloroplast DNA not only contributes to several aspects of normal functioning of cells, its genomics has opened up an area of genetic engineering for crop improvement and environmental sustenance. Thank you.